Scientific breakthroughs often arise from the simplest of inspirations, and in this case, nature itself has provided a promising lead in the fight against Alzheimer's disease. Researchers at Scripps Research have uncovered a way to harness the power of carnosic acid, a natural compound found in rosemary and sage, to create a potential anti-inflammatory drug that could significantly impact the treatment of this devastating condition. By stabilizing and enhancing the properties of carnosic acid, scientists have developed DACCA, a compound that not only crosses into the brain but also selectively targets areas affected by inflammation, offering a new and highly precise approach to slowing or even reversing cognitive decline. Inflammation has long been recognized as a key contributor to Alzheimer's disease. As neurons become damaged, inflammatory responses in the brain intensify, leading to the destruction of synapses, the vital connections between nerve cells that enable learning and memory. The loss of these connections is closely linked to the progression of dementia. The remarkable aspect of DACCA is that it is activated by inflammation itself, meaning it only becomes fully active in areas of the brain that need treatment. This highly targeted approach reduces the risk of side effects and ensures the drug works where it is most needed. The results from studies in mouse models of Alzheimer's disease are particularly exciting. Over 3 months of treatment, DACCA not only preserved synaptic density, but actually increased the number of synapses, effectively restoring brain function to near-normal levels. Behavioral tests showed significant improvements in learning and memory, suggesting that the compound does more than simply slow down the disease. It actively repairs cognitive damage. In addition, the drug reduced the accumulation of amyloid SS plaques and phosphorylated tau aggregates, 2 hallmarks of Alzheimer's pathology. This multifaceted action makes DACCA an especially promising candidate for future treatments, as it simultaneously addresses inflammation, oxidative stress, and protein misfolding, all of which contribute to the disease's progression. Another key advantage of DACCA is its stability. While pure carnosic acid oxidizes quickly, limiting its effectiveness as a drug, DACCA is designed to be stable during storage and is efficiently converted into active carnosic acid in the body. This means it delivers a higher and more consistent dose than taking carnosic acid directly. Additionally, safety studies indicate that the compound is well tolerated, even reducing baseline inflammation in the digestive tract. Given that carnosic acid is already recognized by the FDA as generally regarded as safe, DACCA may have an easier path through clinical trials than entirely new drug compounds, potentially accelerating its availability to patients. Beyond Alzheimer's, DACCA's ability to combat inflammation suggests potential applications for other diseases linked to chronic inflammatory processes. Conditions such as Parkinson's disease, type 2 diabetes, and cardiovascular disease could all benefit from a treatment that targets oxidative stress and inflammation at a cellular level. Furthermore, the compound could be used alongside existing Alzheimer's therapies, possibly reducing side effects associated with amyloid-targeting antibody treatments and improving their overall effectiveness. The discovery of DACCA is a testament to the power of interdisciplinary research, where chemistry and neuroscience converge to transform a natural compound into a sophisticated medical solution. It also underscores the growing recognition that neurodegenerative diseases are not caused by a single factor, but rather by a complex interplay of inflammation, oxidative stress, and protein aggregation. By addressing these multiple aspects simultaneously, DACCA represents a promising step forward in developing a more effective treatment for Alzheimer's disease. As research moves toward human clinical trials, the hope is that this innovative compound will translate into real-world benefits, providing new options for patients and their families. Alzheimer's disease affects millions worldwide, and while current treatments offer only modest benefits, DACCA's ability to restore neuronal connections and combat multiple disease mechanisms raises the possibility of a more profound and lasting impact. If successful, this natural-inspired breakthrough could mark a turning point in how we approach neurodegenerative disorders, offering renewed hope for those affected by these challenging conditions.